Boehringer Ingelheim Vetmedica Incorporated is a global manufacturer of biological vaccines for different species of animals: pigs, cows, horses, cats, and dogs. For more than 30 years, Boehringer Ingelheim's veterinary vaccine manufacturing operations have been located in St. Joseph, Missouri. And in 2009, the company added a second vaccine manufacturing facility in Fort Dodge, Iowa. Each year, over 1 billion doses of vaccines are produced by more than 1,100 employees in multiple shift operations and distributed to more than 50 countries worldwide. Driven by the on-site research and development teams for new vaccines and supported by the International Regulatory Affairs, Quality and Compliance, Logistics, and Plant Services groups, manufacturing operations ensures that both existing and new high-quality products are distributed to these markets in a timely manner. This process ensures Boehringer Ingelheim's mission to deliver leading and innovative solutions to prevent, treat, and cure diseases in livestock and companion animals. Vaccine production consists of 3 major steps: antigen production, final vaccine production, packaging and labeling. Let's review these 3 steps. Antigen production. Antigens in veterinary vaccines can typically consist of viruses or bacteria. They are produced in complex media, liquids that contain nutrients such as carbohydrates, proteins, and vitamins, which are essential for the replication of the organisms. The powdered media components are weighed, added to a medium preparation vessel where high-quality water is added to dissolve the powder, and then it is sterilized by either membrane filtration or heat in an autoclave or vessel. Let's take a more detailed look at bacterial antigen production. There are 2 different kinds of bacteria: aerobe bacteria, which require oxygen for growth, and anaerobe bacteria, which do not tolerate any oxygen. More than 40 of these organisms are grown in fermenters up to 5,300 liters in size, with a total capacity of over 25,000 liters. In order to produce antigens on a large scale, multiple passages of growth occur, from the storage of seed material in small vials to initial growth in small glass or stainless steel containers, to the final large-scale fermenters. In order to avoid contaminations or cross-contaminations, the initial passages are carried out in isolators or biosafety cabinets. Under strictly aseptic conditions, the bacterial cultures are transferred from one container to another and then incubated under the appropriate conditions for the individual organism. Further transfers into the larger stainless steel fermenters occur through steam-sterilized closed connections and are supported by sophisticated computerized systems. This assures an automated process with consistent and high-quality control under good manufacturing practice conditions. Once the bacterial fermentation is completed, the suspension is transferred to the downstream areas for further processing. Depending on the product, this could be either a concentration step, where the suspension is passed through hollow fiber membrane filters to remove liquid and thereby concentrating the antigen; a centrifugation step, to separate the bacteria from the liquid; a filtration step, to remove solids from the suspension; or an inactivation step, where the live organisms are chemically inactivated under controlled conditions. Finally, the antigen harvest material is stored in plastic or stainless steel containers until it has been tested and released successfully for further processing in the final vaccine batch. Now let's take a closer look at viral antigen production. Since viruses are not able to replicate on their own, they have to be produced in host cells such as mammalian cells or insect cells. At BIVI, we routinely maintain about 20 different cell lines for use in the production of more than 50 different viral antigens. These cells are stored frozen in liquid nitrogen at temperatures well below -100 degrees Celsius. To start the cultivation, the cells are removed from the liquid nitrogen storage container and transferred into growth flasks under sterile conditions. Some cells require a surface for replication. Those are grown in rotating plastic containers called roller bottles. Other cells can be grown in suspensions, and we use glass vessels with an impeller for mixing. The cells are routinely sampled and can be monitored under the microscope to assure the highest quality and the proper time for transfers. Since cells typically replicate only once every 24 hours, the process of achieving final production scale can take up to several weeks. Once the cells have been passaged and expanded to large quantities, they are transferred into bioreactors up to 3,200 liters in size. After the virus seeds are added to the cells, they infect the cells, replicate within the cells, and finally disrupt the cells to be released into the medium where they can be harvested. At Boehringer Ingelheim, we use stainless steel vessels or disposable bioreactor systems for virus production. Some vessels contain stainless steel springs or small polymer beads as a surface for the cells to grow on. All other activities, including downstream activities, are similar to the bacterial processes. The bioreactors are highly automated, using computerized controls for online and offline monitoring, and managed by specially trained technicians. 3 of the viruses made in St. Joseph are human pathogens and are manufactured in a separate so-called BL3 suite. Special personnel protection, including vaccination, ensures safe handling of these products. Similar protection exists in Fort Dodge for handling live rabies virus. Final vaccine production. There are 2 major kinds of vaccines: modified live vaccines, which contain live bacterial or viral cultures, and inactivated vaccines containing chemically inactivated organisms. During vaccine formulation, one or several bacterial or viral antigens are combined with other necessary vaccine components, followed by the transfer of the bulk liquid in sterilized transfer lines to the filling room. For live vaccines, the active, live organisms must be preserved during the shelf life of the product. Sterilized glass vials are filled with vaccine at a precise volume which is checked for accuracy several times during the filling operations. High-speed fillers in Fort Dodge are mainly used for companion animal vaccines, whereas high-volume fillers for livestock vaccines are in place in St. Joseph. The entire filling process is carried out under very strictly controlled aseptic environmental conditions, with sterile laminar airflow in the entire filling line. The partially stoppered vials are manually transported and loaded into the freeze dryer. This highly sophisticated computerized equipment allows a controlled freezing and water evaporation process. The remaining dry vaccine cake allows long-term storage and can be rehydrated with a liquid prior to use of the vaccine. At the end of the freeze-drying cycle, the vials are stoppered automatically under vacuum and then removed and transferred to the capping operation where the final seal is applied. At Boehringer Ingelheim, up to 3 million doses of freeze-dried vaccine can be produced per day in this fashion. The filling process for inactivated vaccines is very similar. However, these vaccines are typically filled in plastic bottles. The bottles are pre-sterilized by irradiation. In addition to vaccines filled into glass or plastic bottles, some vaccines are filled directly into pre-sterilized syringes. Packaging and labeling. Packaging and labeling at Boehringer Ingelheim is state-of-the-art. From the highly innovative machines to the technically trained workforce, the department is the final step in the manufacturing process. In 2012, BIVI packaged approximately 32 million units, equaling 102 million bottles and syringes in 900 different packaging presentations for over 50 countries. Batch sizes range from as small as 15 units up to 200,000 units, in sizes from 1 milliliter through 1,000 milliliter bottles. The packaging and labeling department employs over 100 team members across 3 shifts. Each of the 18 packaging centers is designed for specific operations that include labeling, cartoning, bagging, tray loading, flow wrapping, auto cartoning, and manual packaging processing. Each product is required to include labels, inserts, display, and meet a number of mandatory requirements to help track batch and lot numbers before it can be distributed to the customer. The packaging and labeling team is responsible for ensuring each product has met the requirements, and in order to do so, has several checks in the packaging process. Not only is each product manually monitored by operators, BIVI also uses sophisticated technology to complete tasks such as thermal imprinting lot numbers and expiration dates on the label, as well as utilizing vision systems to ensure every label has exactly what regulating agencies require. Packaging personnel inspect every bottle as the bottles are prepared for subsequent cartoning, and operators and inspectors reject bottles that do not meet stringent quality standards. The unit cartons, which is what most end customers see on the shelves, also require information to be displayed, such as the lot number, expiration date, and barcode, to meet regulatory requirements. And similarly to the labeling operation, a laser system and technically advanced vision system are utilized to not only imprint the information on the carton, but verify the information is correct. The one departure from the above concept is the automated cartoning line, affectionately known throughout the corporation as the Autobahn. This technically advanced machine is named after one of the fastest routes traveled in the world because of the speed, quality, and efficiency. The Autobahn winds 140 feet throughout the St. Joseph packaging facility and is capable of labeling, cartoning, case packing, and inspecting over 400 different products produced by the company at speeds of 250 units per minute. These numbers represent about 80% of all the biological products manufactured in St. Joseph. The equipment supporting the Autobahn includes many computers, cameras to monitor various product attributes, computer-controlled motors, lasers, weight-checking units, and hundreds of sensors. The Autobahn is one of the most technically advanced packaging lines in the animal health industry and utilizes innovative laser technology to package product. Upon completion of the packaging process and work, the batch record and remaining labeling components return to the Label Control Center for reconciliation. Material handlers move the finished product to a cooler, pending final release to distribution. Once the Label Center completes reconciliation of the record's components, a thorough departmental record review is completed. When department reviewers complete their record review, Quality Assurance receives the record and performs a final distribution release review. When QA releases the product, it is sent to BIVI distribution partners, who then manage shipment to the customer. It is our mission to supply the global markets on time, in compliance with all regulations, and strive towards a zero-defect culture through our business process excellence initiatives. Excellence in technologies and tight controls in our manufacturing process guide us toward this goal and deliver value through innovation to our customers.